Hello everyone, welcome to Get My Class. So let's solve this question which says PCR is not used in option A detecting site-directed mutagenesis, option B detecting antibody, option C detecting viral antigen, option D confirming infection by detecting particular marker. Now let us see what is PCR first. So we have to actually look for the one which is where PCR is not used. But before we see that, let us talk about what is PCR. What do you mean by PCR? PCR is nothing but polymerase chain reaction. What is PCR? Polymerase chain reaction. And why are we talking about polymerase chain reaction? Because here a series of reactions will occur so as to amplify, you can say a particular nucleic acid. Okay, amplified nucleic acid. When we say nucleic acid, we are talking about RNA and DNA. So PCR can detect only RNA and DNA. Okay, it can amplify only RNA and DNA. Okay, further based on that, further detection can be done. So one of the important, let's say, application of PCR is in detection, or we can say in detection of viral infection. Okay, viral infection. So now many people are worried about corona. So corona for everybody, they're using this PCR to detect the viral infection because they can, even when the sample size is very low or very less, still PCR can be used because the genetic material of the virus can be amplified. Okay, how can we find out the viral infection? By looking for the genetic material. Genetic material. Okay, and this genetic material can be amplified and then further can be finalized that, or you can say concluded that this, yes, this virus has been responsible for this infection. Okay, let us see the application. So option A says detecting site directed mutagenesis. Yes, PCR is applied here where specific, you can say specific sites are mutated, and further the same thing will be amplified and checked if the mutation has been caused or not. Okay, specific sites will be mutated. So we are talking about with respect to the DNA. So that can be detected by PCR, but here we have to look for the one which is not used, where it is not used. But option A, it is used. Option B, detecting antibody. So PCR is not used for detecting antibody. So this is not used. Why? Because this can be done by various methods like ELISA, where enzyme-linked immunosorbent assay can be used for detecting antigen also, and also detecting antibodies. So like that, you have many other methodologies by which you can detect antibody, but not PCR. And this should be a correct answer because we have to look. We are looking for where PCR is not used. Then option C says detecting viral antigen. Yes, it is used for detecting viral antigen, as I told you. First, we will see whether the nucleic acid for viral antigen is like, nucleic acid for that particular viral production is present in the sample, and then that will be amplified. Okay, and further we will know the presence of virus or not, whether it is present or not. So hence this is correct statement because PCR is used here, but that is not our answer. Option D is confirming information by detecting particular marker. Yes, certain genes, they have particular marker genes, okay, or marker areas, or you can say marker sequences, which will show that there can be a particular infection. Okay, and how can we detect? That has to be amplified. That marker has to be amplified. And further, we can say that you can confirm whether the infection is there or not. And that can be, again, for that PCR can be used, but that is not our answer. We are looking for the one where PCR is not used, that is option B. So what is the correct answer? The correct answer is option B. I hope you understood the explanation. Thank you.